Hello everyone. Hello everyone and welcome to this audio podcast. This audio podcast is part of the Prompt series. This audio podcast is part of the Brawn and Rumble. This audio podcast is part of the Atomic Headline, and this audio podcast as part of a Storm Robotics production. So in this audio podcast, looking in a little bit around one of the sort of more one of the sort of main sort of microbiological molecular investigations that are used or have been used retrospectively and are still used and are still used that of polymerase chain reaction or PCR, and this is aligned to the amplification methodologies in microbial diagnoses. So PCR is a method replicating a piece of DNA in a logarithmic manner, such that one copy of DNA becomes two copies becomes two copies or becomes four copies, etc. And if ribonucleic acid or RNA is to be analyzed, a complementary DNA copy of the genomic sequence is first produced by reverse transcriptase prior or before to amplification. The methodology uses a thermostable polymerase and changes it in temperature. Changes in temperature require a thermocycler. So just to iterate here, listeners and viewers, that the terminology and the terminology aligned to amplification methods in microbiology diagnosis is where, for example, as said, that a piece of DNA is replicated. A piece of DNA is replicated in a logarithmic manner, such that again one copy becomes two copies, etc., etc. So, therefore, the Therefore, then, if the RNA is analyzed, the ribonucleic acid is analyzed. It is then where that is where the methodology forms its name, that it basically amplifies the sort of the DNA and RNA, hence the basis of the amplification methodology. And in a PCR, there are three main stages involved in each cycle of replication. There is firstly the denaturation of the DNA. Then there's the annealing of the oligonucleotide primers to the separated strands. And then thirdly, extension of the primers to replicate the complete region of DNA being amplified. The analysis of PCR products is where PCR is a means of amplifying target DNA, but the product must then be analyzed. Analysis may be carried out by a number of methodologies. So, for example, if historically, for example, ethidium bromide or fluorescent stain gel, for example, may be utilized to check the size, but not the specificity of the PCR product. Secondly, a further PCR reaction with a second pair of primers. A second pair of primers referred to as nested PCR, and then analysis by agarose gel for electrophoresis. Then, thirdly, a dot or southern blot is utilized. Fourthly, a plate bead solid phase hybridization and detection. And there are various commercial sort of kits that can be obtained from from organizations, for example, where laboratory practitioners can form the solid phase hybridization and detection. And then there's the real-time PCR, which is not so new now, well, not so novel now, but it is a methodology by which the accumulation of products can be assessed during the amplification process, utilizing intercalating fluorescent dyes or sequence-specific labeled probes. The light cycler, there are two sort of systems that are used by microbiology investigators. For example, have been referred to as the light cycler and the Techman, which are systems that are use this facility, and these will make a major impact on diagnostic microbiology. These have made impact on diagnostic microbiology over the years. So that draws this audio podcast to a close. The next audio podcast will look into a little bit around nucleic acid sequence-based amplification, or NASBA. So this is an audio podcast now draws to a close. This audio podcast is part of the Prompt series. This audio podcast is part of the Ball and Rumble. This audio podcast is part of an Atomic Headline, and this audio podcast is part of a Storm Logistics production.